The title of this on this slide is using historical analogy to gain insights and potential effects of future climate changes for the Chesapeake Bay ecosystem. I could write that title at the end of the talk. If I were to write it at the beginning in terms of exactly what I was going to find, it would have perhaps read a little differently, and that goes to Bill's comments about what we normally think of in terms of restoring the bay restoration. We all know that we're looking towards ecosystem-based fisheries management approaches to help us do a better, perhaps more realistic job of managing our fisheries. So the talk is put into that context, looking for factors that connect different components of the ecosystem that we need to accommodate when we make our policy management decisions. So many of you probably already know turns out there's a strong correlation in terms of the number of young of the year fish that are produced in Chesapeake Bay every year among anadromous fish. And that's what you see here. This is a scatter plot matrix. So if you want to read this, this is striped bass on the y-axis. In this case, that's white perch underneath the striped bass tail on the x-axis. And you can look at blueback herring versus striped bass or any other species listed here in that way. What you notice is the anadromous fishes, most specifically striped bass and white perch, are positively correlated. These are the young of the year abundance in any given year. The years aren't listed, of course. Just when one's high, the other's high, and when one's low, the other's low. What we also see is these shelf-spawning fishes, and it extends more to the menhaden spot, but they're the two strongest, also have a positive correlation among themselves. Spot and menhaden both spawn on the shelf, and somehow their larvae end up migrating into the bay when they aren't, we'll just say, aren't strong swimmers at that point. Various mechanisms might be applied, like counter-current flow, where they go up and down selectively to take advantage of in-current, up estuary currents that are tidal in nature. But very importantly, these two species groups turn out to be negatively correlated. Striped bass and Atlantic menhaden, for example, in that box. Every single year since we've been measuring throughout the bay, no matter what data set we look at, no matter what survey we look at, the VIM surveys, DNR surveys, power plant surveys, says the same thing, that these species are negatively related, and they have been for over 40 years. Today, for simplicity's sake, that the multi-species nature of this correlation was detected through principal components analysis. But it's so strong you can see it in the raw scatter plots. We're going to focus on that Atlantic menhaden versus striped bass box right here. So instead of using PC scores, we're going to use as our response variable something that is obtainable to everybody through the web. You could literally just download the menhaden juvenile abundance index from Maryland DNR and the striped bass, divide one by the other, and take the log of that ratio. And you take the log so that it's normally transformed or normally distributed. That way you can use it with normal correlation studies or regression studies. So this basically goes over our methods that I just outlined for you in terms of the sea bass index we use here. Sea bass, of course, is Chesapeake Bay anadromous shelf spawning index. You might see sea bass RBI here because I want to make sure that I keep track. We're using the ratio-based index here as opposed to a principal component score. This is what the principal component score looked like, and you can see this in the publication by Wood and Austin in the Canadian Journal of Fisheries and Aquatic Sciences in 2009. The correlation between this index and the ratio-based index that we're using today is 0.9. So there's no problem in using that ratio-based index. There's only advantages. So we're going to start to look at, investigate in this talk what in the world's going on here. It's very important to understand if there's this multi-species complex, and Wood and Austin did a good job, I hope, of convincing folks that this relationship doesn't have to do with fisheries and a simple fisheries exploitation rates. A simple explanation is that menhaden, spot, striped bass, white perch, and the herrings all have experienced very different fishing efforts. You can't put those fishing efforts together and get the signals you see. There's a longer story if you want to look up that journal article to be told, but I'll leave it at that. So if we're trying to figure out what in the world's causing this multi-species pattern in Chesapeake Bay, that of course affects the coastal populations of our economically and ecologically important fish like menhaden and striped bass. One of the first things we can take advantage of is the Bay program's plankton data set. If these fish are being driven by factors like climate factors, which is what Wood and Austin suggested as a principal candidate, because the sea bass pattern is shown to operate synchronously throughout the entire bay, then you should see a response from the plankton. Moreover, the plankton are actually the prey for these fishes in their very early life stages, and we'll talk a little bit more about that later. We're going to backtrack then to figure out what's going on here and looking at estuarine hydrography. Because of climate should be a forcing mechanism, you'd see it in the hydrography, and that might affect the plankton, that might affect the fish in a trophic relationship, or it might affect both of these just coincidentally. And then of course weather should be affecting the hydrography. And then we're going to ask, because teleconnections or large-scale climate connections throughout the hemisphere. These are indices, they're sea level pressure patterns that are shown to affect weather in two very different parts of the world. For example, El Nino, or 3 in that case, or 4. So we're going to look at candidate northern hemisphere teleconnections to see if there's a connection between, if there is indeed a weather pattern caused behind the sea bass pattern, whether a teleconnection might be responsible for that. And that's because teleconnections often provide lead time. They often have momentum or repeatability, repeating patterns through months or years. And so if you can, in terms of weather, a long-term prediction, if you want a long-term prediction, you look at these large-scale sea level pressure patterns or teleconnections. That's how our 30, 60, 90-day lead forecasts by the Weather Service are actually generated. And then we want to see if, in the end, this might have an effect on fisheries landings, because that's the holy grail. Not only understanding the patterns in our ecosystem, but also understanding what their effects are, and then getting some policy implications out of that information or those investigations. So why do we start where we start? It's because it's very important to note that both of these fish species, or fish species groups, represented by Atlantic menhaden, which is a self-spawner, and striped bass, which is an anadromous fish, both of them use common regions of the estuary. I call it the oligohaline, mesohaline transition zone. They use those reaches for their primary nursery areas, and it's thought for fish that recruitment is usually largely determined within their very early life stages, weeks to months after birth, after hatching, I should say, in most cases. So this is from the Potomac River Atlas, and you see that same bend is utilized by both species as a nursery area. But so they should have something in common so that they both react synchronously, but they react oppositely. So they need to have something that's opposite. And indeed, the shell spawners are coastal spawning. They spawn in the winter to early spring, and they rely upon some sort of up estuary migratory mechanism to arrive in these, I'll just call it OMTZ zones, from March to June, where they begin, for Atlantic menhaden specifically, they begin feeding, transition from pickers of zooplankton when they're very young to filter feeders on phytoplankton. And they remain filter feeders for their entire first year of life within the estuary. While the anadromous fish are actually deposited there as eggs, they're retained somehow, and they develop there. They rely in those same months from March to June upon zooplankton species. So we're going to look at the plankton community, as I said, either as an indicator of climate, forced habitat changes, or possible predator-prey linkage, or perhaps a little of both, as often is the case in nature. And we're going to use plankton data from the stations spanning this OMTZ zone. You see the stations there. We're only looking in Maryland because microzooplankton were thought to be very important, perhaps, to both trophic linkages within the plankton community, but also perhaps for Atlantic menhaden, which would start as they're very young, probably filtering or preying on those zooplankton species first, and perhaps filtering them too as they ontogenetically change their gill rakers before they feed entirely on plankton. So what we did was we summed, we took the major phytoplankton taxa at all these stations. Essentially what we did was take them all, sum them all for all those stations, aggregated them. And so I had one time series for each of these major taxa, and we decided what a major taxa was by essentially, there were lots of them. We didn't need a gazillion of them. We needed the major taxa to see if there was a major phytoplankton or plankton signal. So what we did was make sure that any taxa that didn't have at least one organism present in every single year, because it could come from any of these stations. If you had none in any single year, you weren't a major taxa, is what we decided. And so we had one single time series for all the major taxa. And here are the major taxa that resulted. But you see there's a little bit of an imbalance in terms of taxonomic identification, and that's because that exists in the data set for one thing. There are some more specific taxa here, like Acartia and Bosmina, because we know, for one, they were counted well, and they're important to fish. So we kept them in there at that specificity. So we have the phytoplankton, the microzooplankton, and the mesozooplankton, including the jellyfishes in this data set. And we decided to run a principal components analysis because that essentially will extract the most important multi-species patterns in the data for this multi-species plankton data set. In the end, this is what you would expect. If this were a completely random data set with no signals, all of these eigenvalues would be here at 1. So what you see is a non-random data set. It's good. We have a very strong first few principal components, but we're going to knock them all out except for number 1. And that's because number 1 turned out to be very strongly correlated with the sea bass pattern at 0.88. Very significant. It accounts for 19% of the variance, though. The first 3 would have accounted for about 50%, and I think it's 47%. So there's 70 variables here. Now you have 3 variables counting for 47, and you have the top one, which is correlated with sea bass, at 20. So what we're going to do is throw out all the others and just look at that first one, because we're trying to figure out, we're using the plankton only as an index of, to answer a question, primary question, are other components of the ecosystem varying with the fish? It so happens they are prey, but we want to know that first question first, because if it's climate, plankton would be varying with them. So here are the loadings. The menhaden should be feeding on these fish at that time. Sorry, those plankton. And the striped bass should be feeding on those plankton at that time. We can talk about that later in the discussion. It's mesozooplankton versus phytoplankton, essentially. They're weighted oppositely on PC1. So now we asked, is hydrography a forcing mechanism here? And so we looked at, just like we aggregated across the entire OMTZ of the bay for plankton, we did that for water temperature and salinity for March, April, May, and June. And then we also looked at bay flow from March to June, from January to June, because we know flow might affect salinity before you actually are looking at the plankton. And the plankton weren't counted in January, so we couldn't use them that early. We could only use March through June. And then we looked at the year before, because there's a chance that the year before might affect salinities. I think Dave Kimmel in one of his papers said there's evidence that flow the year before might help the zooplankton, so they might have resting eggs for the next year that come out real strong. And indeed, that 18-month prior flow is actually a better predictor than just the January to June, which is a little bit better than March to June. So here's what we look like. Here's May salinity. The sea bass pattern are those steps. Plankton PC1 are the dashed lines, and bay flow are in the blue lines. Clearly, really, really strong correlations. So now let's look at regional weather patterns. And what we see, the regional, in fact, the continental weather patterns, spring temperature from March to May is negatively related to Plankton PC1. Sorry, that's precipitation. Negatively related to Plankton PC1 and negatively related to sea bass pattern as well. There seems to be a slight positive correlation with temperature, but it is fairly low in the 2 to 3 range, 0.2 to 0.3 range in terms of correlation. that may either be real or it may be that temperature is that way when you tend to have dry conditions. So what's forcing this? We're going to step into sea level pressure and atmospheric circulation. This is the atmospheric circulation, or the sea level pressure, sorry, as it pertains, as it is correlated with the sea bass pattern from 1966 to 2006. In which what's important to note is there's essentially a high pressure anomaly that exists when sea bass is positive, which is when the menhaden would do well, and a low pressure to the north of the bay, essentially, when the coastal spawners are doing well. And we'll hear a little bit more about that. This is what the strongest years of striped bass recruitment look like, according to the Juvenile Abundance Index from Maryland DNR. This is the top quartile years. This is what the signature looks like. And what you see is this classic Cape Hatteras low here. Generally low pressure over the Atlantic, high pressure to the north, Canadian Maritimes. If you can see the stream trajectories, this is surface airflow. You're getting cold air coming down and combining with those northeasters. Typical pattern that we can see in the wintertime. Blocking high, it could easily block the low from moving up north, and so it could sit here a little longer and give us a big snowfall. That's what happens quite often. So that seems to explain flow positively. When you have menhaden in their first quartile, you get the opposite condition with stronger than normal high pressure, which pushes the storm track to the north, because wherever there's a low anomaly here, it means the storms are traveling this direction. And what you see is the wind is coming up from the southwest into the bay, so you're talking about warmer, drier conditions. So now let's look at the teleconnections. Is there any teleconnection here that might be at work? We saw these large-scale patterns, very consistent. There's some hope. So when I looked at a bunch of teleconnections, Pacific Decadal Oscillation, North Atlantic Oscillation, Southern Oscillation, or El Nino, and the Pacific North America pattern, and this thing that's relatively new, the Atlantic Multi-Decadal Oscillation, that's the one that popped out as being significantly correlated. All the others were about zero. This is like El Nino in that it was first discovered as an oceanic feature. It means what happens is the Atlantic is warmer than normal whenever the AMO is positive and vice versa. And what we know, previous studies in the last 5 years have shown that there's a real precipitation signature. They've talked about the drought during a positive AMO year. But if you look closely, the opposite happens right at the coast. It's the kind of thing we've been seeing where that precipitation is tight to the coast. That's because perhaps of northeasters, and we can talk about the climatology behind that later. But here's also the surface air temperature, of course not the water temperature you saw there earlier. This actually extends the land to see the big picture. And here's sea level pressure resulting from the warm waters. Whenever you have warm waters, the air lifts, you get low pressure. So this translates, this oceanic feature translates to weather that way. Here is the correlation with sea level pressure with the sea bass pattern. If you notice, it's pretty much exactly the same, but opposite. That's why the AMO was negatively related to the sea bass pattern. In other words, positive is good for striped bass, negative is good for Atlantic menhaden. So the question is, does this have anything to do with fisheries landings? And the answer is yes. I'll try and fly through this. For the Atlantic menhaden, there's been a strong trend in their technology usage from the 1800s. So what we did was first detrend the harvest. Then, because the AMO is influencing the recruitment of the fish, we took the harvest and we lagged it by the average age of catch, 2 years. So that now we're in sync with when the AMO should be influencing harvest 2 years later, because these are 2-year-olds that we're catching primarily. When you compare those two time series, you get the triangles, sorry, that's straight bass. You get the open circles, and then the AMO are these sticks coming down from the top, stalactites, I believe. What you notice is a lot of the interannual variance is not accounted for over the last 100 years. But this pattern, by eye, is remarkable. And in fact, the correlation, once you take this low as smooth and compare it to the Atlantic menhaden harvest, is 50%. What you see in this light are the unadulterated juvenile abundance index values for Atlantic menhaden. So Atlantic menhaden, young of the year, are correlated with the harvest 2 years later. Remember we lagged it. And with the AMO pattern for 100 years. The same thing is true with the striped bass. And again, you're talking about 50% of the variance. Here's the striped bass catch. It's lagged 8 years because the average age of the coastal fish is 8. And these were just Chesapeake Bay harvests through time. These are striped bass harvests are for the Atlantic, mid-Atlantic states and the Chesapeake. And what you see again, these bars, these dashed bars, are the juvenile recruitment index. The triangles are the harvest, lagged 8 years. And then the rest is either the lagged or the raw AMO scores. So, take-homers. There's a lot of take-homes from this. I won't read from this slide, but essentially this gives us an opportunity to, number one, realize that it's not just about manipulating fishermen to restore our fisheries. While we'd like that for the case, at least everyone but the fishermen, it just doesn't seem to be the case. Natural variability is very important. The Chesapeake Bay is in the mid-Atlantic area. It's very vulnerable to wide ranges of weather conditions during the spring, which itself is a very tumultuous time. It can swing widely. You would expect the living resources to respond, and they seem to. Again, we should probably talk and discuss what this means, but it has implications for the looming specter of climate change, as I like to say, for how we manage our stocks. Perhaps we can't control it, but perhaps we now can anticipate wide changes and both protect the fish and also perhaps exploit the fish at higher rates when we know it's safe to do so. So that's the big picture, and hopefully there'll be, if folks find this holding up to the challenge of peer review as time goes on, we'll be in discussions with policyholders, and they'll be asking the questions about the science and maybe what we would advise for them to do. So with that, I'll take my leave or take any questions you have for me.
Thank you.
Thank you.